Thank you, Steve, and thank you everyone for coming to this talk in the afternoon. And I'll be talking now about modeling hybridization between species and how do we reconstruct that from sets of gene trees. In fact, in the previous talk, we heard the words borrowing and contact. That's the equivalent of hybridization between languages. Okay? So there are two main parts to my talk. First, I'll start talking about gene trees, how we compute their probabilities when there is hybridization happening, and how we infer evolutionary histories of species from these gene trees. And then in the second part, I will go to, if we have the full genome, whole genome sequences, how do we annotate these genomes to find regions in these genomes that have undergone what we call introgression, which is some segments of DNA get fixed in these genomes. So I'll start first with talking about hybridization, phylogenetic networks, and how we infer them. So in the morning, we heard about incomplete lineage sorting. So here we have an example, again, of a species tree. I'm a computer scientist, but I know that these bell peppers are not different species, but it's just an illustration. So this is a species tree. Just imagine these are 3 species there. And we have two illustration of 2 gene trees growing inside this species tree. If you look at the blue one, again, it agrees with the topology of the species tree. If you look at the red one, it disagrees with it. Okay? And I will show you actually later an exact formula for telling us exactly the probability of this happening. So when we run into incomplete lineage sorting, again, we will be having collection of gene trees that disagree with each other and with the species tree, and we want to infer that species tree. And another factor that can cause gene tree incongruence is hybridization. Okay, so when we have hybridization, imagine we have the gene flow happening from one species to another, from one population to another, and when this happens, now if you look at gene copies in this hybrid population, you can trace the evolution of some of genes to the left parent. I'm going to call it left parent, or some of them are going to go right. So here in this case, if you look at the blue and the red genes, we exactly have the same 2 topologies that I showed before, exactly the same incongruence between them, but incomplete lineage sorting is not playing a role here. This is because of hybridization. Now where the picture gets a bit more complicated is that incomplete lineage sorting, what happens if incomplete lineage sorting and hybridization both happening at the same time? So now in the previous, just to show you, in the previous one here, the incongruence between the 2 trees, if you look at this simple case, if you look at this incongruence, I take one gene here, it attaches to the right parent. I take another gene, attaches to the left parent. So you can imagine now one principle to reconstruct or infer this hybridization is just look at that incongruence. Where is it going? Once with the left parent, once with the right parent, then you can imagine there is hybridization happening between these 2. But the problem is that if incomplete lineage sorting is happening at the same time with hybridization, then if you look at this gene tree, for example, and you strip out that network from the picture, you will see that this gene tree is actually giving us a similar shape to the blue gene tree. Okay? So here hybridization happening, but incomplete lineage sorting is actually hiding or masking the signal for hybridization. So the picture gets much more complicated here. So why do we want to focus on hybridization? This is a review from 2005 that basically stated that about 25% of plant species and 10% of animal species are involved in hybridization. And I do believe actually that these estimates are going to go up when we have methods to detect them, because again in the morning we heard about concatenation and so on. One of the interesting things about concatenation, even if you have hybridization, is going to give you a tree. And many analyses are done with these kind of methods. And also in this very review by James Mallet, he actually showed a picture of where incomplete lineage sorting can happen. So here looking at multiple individuals or multiple loci from this species, some of them can trace back the hybridization event. Some of them can take the vertical parent in some sense. So the question here is how do we tease apart the signal of incomplete lineage sorting from hybridization so that we get the true number of hybridization events.
Okay. By hybridization, you mean merging of 2 whole genomes? Or?
Hybridization here, imagine gene flow happening between populations, for example, or species. So when we talk about hybridization here, immediately after hybridization, it's like sexual reproduction, right? So if you have hybridization between 2 diploid populations, then yes, you have an individual all of a sudden that has 50% of genetic material from one parent, 50% from the other parent. But the picture gets much more complicated after that because of selection and because of recombination and because of drift. The percentage doesn't stay at 50%. So now we look at genomes that are mosaics, have parts of these signals of hybridization. And another interesting thing about hybridization is that in some recent examples have been shown in the literature that demonstrate an adaptive role to hybridization. So for example, this is a very recent paper, 2012, in Nature that showed that hybridization happening in butterflies, and it's responsible for mimicry in these butterflies. This is here by a colleague of mine from Rice University, where they showed that there was introgression or hybridization between mice that conferred resistance to the mice from rodenticides. Okay? And the rodenticide that was used here actually is warfarin, which is used also as a drug on humans. So hybridization does happen. There is more and more evidence for it. Traditionally, people think about it happening mainly in plants, but there are more evidence now happening in animals. And again, with the development of new methods and more data, more signals going to be detected. So my interest is how do we reconstruct evolutionary histories of species from sequence data or from gene trees that account for hybridization, and also at the same time account for incomplete lineage sorting. So this is here a general formula that was given by Wayne Madison, and it makes sense. It was given by Wayne Madison in '97, but if you think about it, basically if we have a set of sequence alignments from multiple loci, and we are interested in '97, Madison was talking about the species tree. If we are talking about the species tree under maximum likelihood, we want one that maximizes basically this entity. If we look at this, we have a product over all loci, simply because of the assumption that the loci are independent. And then for every locus, when we look at the sequence alignment for it, you can now sum over all possible gene tree topologies. There is an integration here that I'm omitting because if we want to also account for all branch links, we have to do an integration. And then we have these two terms. This term here is the probability of sequences given a tree, and we know how to compute this. Again, we have seen in the previous talk mention of Felsenstein's algorithm. For example, we'll compute this entity. And this has been our focus because this is what's missing now. For incomplete lineage sorting, if gene trees disagree with species trees only because of incomplete lineage sorting, then this has been worked out, this probability. What we have been focusing on is how do we compute this probability if there is also hybridization as well. Okay? Now, if in the morning we heard about approaches where the gene trees are reconstructed first, and again the talk after this we will hear about gene trees reconstructed first. If we have that, then we can factor out completely this issue of sequences and integration over gene trees, and we can basically focus on the probabilities of the gene trees. Okay? So the question is, how do we compute this probability of gene tree given a species tree? So here in the case of species tree, and then I will move on to what happens when there is hybridization. So here there's no hybridization, incomplete lineage sorting is going to happen. Look at this species tree here on 3 species A, B, C, and I have an example of a gene tree there, A, B, C also. Okay, so gene A is taken from species A and so on. Now, if we look at these two coalescent events, basically the two genes from A and B coalesced at node X, and then we have node Y. And I'm focusing exclusively on topology here. I'm ignoring branch links on the gene tree. There are 2 possible ways to explain that gene tree appearing inside this species tree. Either A, B coalesce on branch 1, so node X is mapped to 1, and then Y is mapped to 2, or X happens here, so A and B coalesce here at this point, and then C coalesces with them. So this is what we call coalescent histories. So there are 2 possible coalescent histories, and if we look at a vector whose size is the number of internal nodes in the gene tree, and we put the entries in that vector are basically the branch numbers from the species tree, we can now represent each coalescent history is with one such vector. Okay, so this vector 1, 2 telling me that one embedding of that gene tree inside the species tree is map X to 1, Y to 2. This is saying X to 2, Y to 2. And there are no other coalescent histories for this gene tree. Okay, because I'm just focusing on the topology. If we focus on time, then the times of these events are going to dictate where these things happen. So once we take these, once we think about this set of coalescent histories, so I'll denote by H sub psi of GT, the set of all possible coalescent histories that can explain the gene tree, then the probability of observing any gene tree given the species tree is given by this quantity. So this is now the probability of a gene tree given a species tree and its branch lengths. And I will illustrate, I will describe what this means, but the main thing to notice here is that this probability now is a summation over all possible coalescent histories. So we are saying what is the probability of the gene tree? It's basically sum over all possible ways of embedding the gene tree and then compute the probability of that embedding. Okay? Now the probability of the embedding is nice to compute because we can take it, it's independent for the branches, so you can compute it for every branch individually and take the product. So what are these terms that are happening, that are used here? Okay? So suppose we have a branch B here whose length is lambda B. So this is a branch in the species tree. Okay? And we are going to focus just on what's happening within this branch and see what is the probability of that thing happening. So imagine here that we have 5 lineages coming into the edge, because we are looking at the coalescent from the leaves towards the root, 5 of them coming and 2 of them exiting. If this scenario happens, then for sure you have to have 3 coalescent events. Right? So to reduce it from 5 to 2, we have to have 3 coalescent events. So the previous term on the formula was, what is the probability that 3 coalescent events will happen within this time? And the second term, which is that we take the product of the two, once we have these 3 events happening, we want them to happen in exactly this way that they give rise to exactly this topology. Right? Because many ways of three coalescent events could happen, but we want the specific ones. So the first term is what is the probability of these 3 coalescent events happening, assuming that 3 did happen. Here, again, we denote by this UB of H is under this coalescent history H, how many lineages are incoming into this branch, which is 5. How many of them exiting would be 2. Sorry, 2. And now we can say these 2 terms that were in the previous formula, WB of H, how many possible ways these specific coalescent events could have happened? There are 3 of them, because we can think about 1 and 2. 1 has to happen before 2, but 3 could happen before 1, between them, or after them. So if we put these in total order, we have 3 possible ways now. Okay? And the other time we have to say, these 3 force the coalescent events to happen in exactly the same way to give rise to the gene tree that we have. We cannot change that. But there's another term, these are 3 out of how many possible ways we could have had 3. And the formula for that basically gives us 180. Okay? So these 2 terms are, if I divide this by this, is going to give me the probability of these specific coalescent events happening. Okay? And the other term in the formula was PUV over time T, is that what is the probability of U lineages entering the branch, V lineages exiting from the branch if the branch had length T. And this formula was derived in the mid-80s, actually, and it has been used in this context and many other contexts. So these 2, this tells me if you have a branch of length T, what is the probability of a certain number of coalescent events happening? This formula is going to give us that. And the previous slide basically told us, for this specific number of coalescent events, what is the probability that the specific pattern of coalescent events will happen? Okay. So these will give us basically the probability of a gene tree given species tree. Some of you might not have seen it in the most general case. So here, this is a three species case, and we heard about it in the morning. So this is human, chimp, gorilla. And if you want to look at there are 3 possible gene tree topologies here. Again, one that puts human, chimp versus gorilla, human, gorilla versus chimp, and chimp, gorilla versus human. Then that formula is going to reduce to these 3 values here. Okay? And these 3 values, again, the main factors that change things are the length of this branch that separate the most recent common ancestors, this and this, the length of the branch, which in number of generations, T2 minus T1, divided by n, which is the population size or the width of this branch. Okay? And if we want to visualize these 3 probabilities, this is the value of the 3 probabilities as this quantity increases. Okay? And this kind of figure and this kind of illustration actually explains why often in phylogenetics researchers don't focus on incomplete lineage sorting, because if your sampling is done in such a way that this branch is very long, you are probably operating in a zone here where this is not really an issue. If you sample enough genes, then the gene with the highest frequency might be indicative of the species tree. Although there is one important caveat here, is that this result is true for 3 species, but there was a seminal paper a few years ago by Noah Rosenberg and James Degnan that showed under certain circumstances the gene tree with the highest probability is not necessarily the one that agrees with the species tree once you go beyond 3 species. Okay? But for 3 species that doesn't arise. The gene tree with the highest probability is going to be the one that agrees with the species tree. Okay. So the question is, what happens when we say, okay, we have hybridization and incomplete lineage sorting. Now, what is the probability of this gene tree growing inside this evolutionary history? So the first thing is that when we have hybridization, the evolutionary history of species is no longer a tree, and we have to go to phylogenetic networks. And this is the definition that we use in our work for phylogenetic networks, and it's very important to give that definition because phylogenetic networks have been mentioned in thousands of papers now, and I think there are at least 20 different definitions probably. Some of them are very close to this. Some of them have nothing to do with this. All of them are called phylogenetic network, but everyone introduces their own definition. And we are all guilty of that. Okay? So phylogenetic networks here are basically the topology of a phylogenetic network is modeled by a rooted directed acyclic graph. And we have basically the root. We have these type of nodes within degree 2 that describe, for example, in our case, hybridization. But if you're thinking about recombination, it will be recombination and so on. And we have the leaves as before with the trees. In addition to this, we have also to have, because we are doing probabilistic inference, we have to have branch lengths on these branches. And in addition, we have also probabilities associated with these nodes that we call inheritance probabilities. So this inheritance probability, for example, for this node here, I have gamma, which basically means that if I'm looking backward in time at genes copied from these species, and I go backward in time, when I'm tracing the evolution of a gene from B, once I get to this node, I have probability gamma of going left or probability 1 minus gamma of going right for that. Okay? So when we do the inference of these networks, we want to get the topology, we want to get the branch lengths, and we want to get these gammas. And if we have k of these nodes, we have to have k of these gammas. Okay. So one of the main concepts in networks that have been used extensively for the last 20 years in terms of how do we reconstruct network, how do we evaluate network, how do we analyze networks, is by looking at something that we call trees induced by networks, or trees displayed by networks, or trees contained by networks. And the concept is very simple. If you have a network, you have nodes of in degree 2. For every possible way of, look at every node of in degree 2 and remove one of the 2 parents from that, 2 parental edges. So you can remove this and this, you will get that tree. If you remove this and this, you will get another tree, and so on. So when you have 2 of them, you have 4 trees in this. And if you have k of them, in general, you have on the order of 2 to the k. It can be smaller than that. Okay? So, but this is the notion of induced trees. And if we have only hybridization, then each one of these trees should be able to capture any of the genealogies in the evolutionary history. Because we don't have incomplete lineage sorting, so every gene you trace it back, it should trace one of the parents and coalesce at the most recent common ancestor. We should not have any noise in the picture. However, the problem is that this notion of induced trees does not capture the full story when ILS is present. Okay? So here, if you look just at this case, I take one copy of a gene from each of these 4 species, and I trace back the evolution of this copy from C and one from B. If you look at B, it goes left, and if you look at C, it goes right. In this case, the tree that should capture this is B is attached to A and C is attached to the species tree. Okay? So this is a case here where the notion of induced trees is not sufficient to capture the evolutionary histories of genes. So for us, the solution that we did for this problem is that instead of looking at a network and its induced trees, which is not sufficient, we convert the network into a special tree. It's one network to one tree, not multiple trees, that we call multi-label tree. And we consider all allele mappings of the gene tree to that MUL-TREE. and then we compute the probabilities. The nice thing about going from the network to tree is that we have lots of techniques that work nicely on trees, from doing parsimony to doing likelihood and doing all these kind of calculations on trees. So representing the network as a tree immediately translates or transfers all these techniques to work on these trees. But there will be just one detail that we have to take care of in this case that I will describe. So I'll avoid that one there, but I'll illustrate what we mean by this multi-labeled tree. So that's a network that's given here, and this is the multi-labeled tree representation of the network. So it's very easy to go from here to here. And this one simple way is just go bottom up. Whenever you see a node like this, just copy the entire subtree under that node, attach it once to the left, once to the right, so you will get this. And you keep repeating, because if you have more and more, you can repeat this process. So what you end up with is one tree that represents this network, but this is not a traditional phylogenetic tree because the leaves are not uniquely labeled. Okay, so this subtree here under the hybridization event is now 2 copies of it, and BC and BC. So the issue that arises now when we have this small tree is that instead of looking at this network and trying to figure out where the gene genealogy, how the gene genealogy traces inside this network, now once I map the alleles to the leaves of the tree, the nice thing is that with a tree there's always a unique path from a leaf to the root. Okay? So what is this concept of allele mappings? When I have this small tree description of the network and I have a gene tree here, if we look at the gene tree, for example, it has, in this case, we sampled 2 alleles, B1 and B2. B1 and B2 could both be sampled from this, from here, or B1, B2, or B1, B2. Okay? So the allele mapping, each allele mapping is supposed to capture one of these scenarios. Okay? So these are all the possible allele mappings that I can find for a gene tree given this MUL-TREE. If we have B1, B2, they can again map both of them to this B, or to each one of the Bs, or to this. Okay, and as we have the C also, the C, because we have one allele, we sampled one allele from C, it can map to this one or to this one. Okay. Now the nice thing about it is that the probability of a gene tree given the network is now the summation over these allele mappings with the probability of a gene tree given this MUL-TREE. Okay? Now this, I showed a formula earlier with the probability of a gene tree given a species tree. Now we had to tweak that. It turned out to be a bit of a challenge, but we had to tweak that so that this probability now applies. Okay? Now why did we have to tweak it? Because if you map B1 to this B and B2 to this B, and then they trace back two different trajectories, then implicit in this, you have to account for the fact that implicit in this, the fact that B1 and B2 did not coalesce on this branch. Right? Because if you don't account for that, you are not getting actually the correct probability. So since I forced them to take different paths, then I have to, basically in the probability, I have to put a term for the fact that B1 and B2 could not coalesce here. Okay? So that was the detail that we had to account for. And to get it done, basically, we have to introduce some mapping that takes care of this very nicely. And that generalizes the probability that I showed before to this. Again, it looks a bit more complicated, but the fact is just about the reason it looks like this is because we have to, again, to account for these copies that came from the same species, but now we are forcing them to take different paths. Okay? And again, in the morning, we heard about the issue of error in the gene trees, and that gene trees are not necessarily estimated correctly. We have 2 heuristics to deal with these things so that when in our implementation, we don't assume that gene trees are binary or the gene trees are correct. You can use gene trees that you have, for example, obtained from Mr. Base and use the posterior to weight these gene tree topologies. Or another thing is that if you are doing bootstrap, then you can contract all the branches that have low bootstrap in the gene trees, turning it into a non-binary tree. And then when you are doing the inference or the computation of probability, you can now find a resolution that is probably better, that the phylogeny is going to constrain. Okay. Now, of course, our main interest is to do the inference of this species phylogeny, which is, in our case, the phylogenetic network. So I have a set of gene trees, this big G, and I want to find the phylogenetic network that optimizes or maximizes the likelihood. And our approach to this is basically to do what people have been doing in the case of phylogenetic trees for decades, which is don't try to solve it exactly without doing any search. We do search in the network space now. Now, in the case of phylogenetic trees, there are many ways of searching the tree space. Here we have to come up with our own way of searching the network space. And the way we do it is we do it in a controlled fashion. We divide the space of networks into layers, where this layer has the set of all trees. This has the layer of all networks with 1 reticulation, 2 reticulations, 3. And one of the interesting things that came out once we started thinking about this is that if you think about trees, if you look at in taxa, the number of branches you can have in a tree within taxa, if it is rooted, is 2n minus 2, right? So you have 2n minus 2 branches in a tree within taxa. That's the maximum number you can have, because you can have non-binary trees. The problem with networks is that in theory, this space is infinite. You can keep partitioning the branches here and making them shorter and shorter as much as you want, in theory, right? So you cannot even limit it in theory, unlike the case of trees. And the major issue that arises here is that once you start going from this layer to this and so on, now the model becomes more complex and it starts fitting the data better. And the trivial answer will always come back as, just take the network that's most complex in the analysis. But we do this analysis here because when we have these operations that allow us to jump from one layer to next, we actually try to weight these kind of operations so that we don't keep taking this with high frequency. And the other thing is that to allow for this search, we have to speed up that calculations. Something I'm not going to show here, which is that we can compute the probability of a gene tree given a phylogenetic network in a much faster way without having to explicitly account for all coalescent histories. So we do a technique that's used on something called ancestral configurations, and that speeds up the computation. And again, the last thing we have to account for is the model complexity. Again, if you want a network that fits the data better, just keep adding reticulations. Okay? And whether it is under likelihood or parsimony, just keep adding nodes and things will get better. And here we experimented with information criteria like a KK information criterion and BIC and so on. But we also devised a new way of inferring these probabilities using cross-validation, where we learn the network from a subset of the data and then use the remaining subset, the test subset, because we know the frequencies of gene trees in it, and we have learned the network, we compute the probabilities of the gene trees. Now we can compare the probability under the model we learned to the frequencies in the test data set. And in fact, this turned out to work well, actually, this cross-validation. So here, this is small. The next slide, I'm going to show the bigger picture of this data set. So here, this is an analysis of a data set of 5 mouse genomes. The mice were sampled from Germany, France, Czech Republic, Kazakhstan, and China. This data set of mice there have been many hypotheses about hybridization happening between them, and the research papers have been actually talking about hybridization between every pair of species there. So it's happening like everywhere. These are results that are coming from our analysis. Here, this is just for exploration. We are showing the top networks with 0 reticulations, then the top with 1, with 2, with 3, and so on. Because again, in exploration, we did not want to stop the method to see what's happening. Based on the cross-validation results, the best result is obtained when we have networks with 2 reticulations, in this case, and it performs much better than AIC and BIC. And this is a better schematic of these results, where we show that for this data set of mice, we actually hypothesize that a lot of the incongruence is due to incomplete lineage sorting, and you don't need tons of hybridization events. 2 hybridization events are sufficient. And this was done using about 20,000 genomic regions from the genomes, because we had to sample them far apart to be independent. What these circles mean is that in our analysis, we find that we can get the target of the hybridization with accuracy, with confidence. We are sure it happened from here to here. But for the source, it's interesting that the likelihood, calculate the values, whether the source is here or here or here, are almost the same. So we basically present this as a consensus of these results. And the same thing with this. We also infer the amount of gene trees that are hybridized, about 6%, and here about 4% between these. And these are consistent with all the findings that are reported in the literature, but this is a much simpler explanation. Okay? We have reanalyzed the yeast data set. Also, again, we heard about concatenation in the morning. This is the yeast data set from a famous study where the sequences were concatenated and a species tree was obtained with 100% bootstrap. Using our analysis, actually, we find that hybridization, an evolutionary history with hybridization and longer branches explains the data much better. Okay. We also have calculations of gene trees when the branch lengths are also incorporated. So everything I talked about now is topologies, but we have also did it for branch lengths as well, which I'm not going to talk about. But it's very important because now, with Felsenstein's algorithm and these methods that we have, you can do the full inference all the way from sequences, in theory. Because once you implement this, this is going to be a nightmare computationally. So in the last few minutes, I'm just going to talk about the second part, which is much shorter, is about how do we use these networks. If we have an inference of network, how do we annotate genomes for introgression? And the point here is that if you look at the genomes, they are basically data that was generated from this stochastic model, a network where genes, we have these gene genealogies are evolving within that, and on top of them we have sequences evolving, right? So we get all these genomes, and unlike the previous part where we were taking regions from the genome that are independent building trees and we want to get the network, now the point is that if the biologist has this data set of genomes and they know there's introgression, some segments of DNA that are introgressed, we want to annotate these genomes. Okay? So our approach is again to use this network concept and the trees, these kind of trees here that are the trees inside along with the allele mappings. And our problem of interest is that if I have the input in part A there, this set of genomes with all the DNA in them, and if I know also something about these trees inside the network, can I predict for every site in the genome, can I predict the probability or the probability for the site coming from which one of these trees and which one of these gene genealogies? Because once I do this, then I can basically threshold this or do whatever I want with it, so that I say that, okay, I believe that the first site evolved down this local genealogy inside this tree. So it's not involved in hybridization, or down this genealogy and down this tree, which basically says it is involved in hybridization. And our approach to this is to combine hidden Markov models with these phylogenetic networks. So now, in this case, since I have two possibilities for the phylogenetic network, either things from B go left or go right, then I can have two rows of states. This is what these two rows of states correspond to. So this is the states where things are going left here. These are the states where things are going right. But even when you go right, you can still have all possible gene tree topologies, right? Because even this red one could have come right and not coalesce here and coalesce in different ways there. So now every state here has a local gene genealogy. In 3 taxa, we have 3 possibilities. If this was 4, we will have 15 states because there are 15 topologies, and we'll still have 15. If this network had more reticulations, then we will have more rows. This is how this model is going to blow up, basically with more reticulations and so on. So now we have, when we have this model, basically switching within these kind of states, switching within this row or within this row corresponds to basically crossing recombination breakpoints and having incongruence because of incomplete lineage sorting. If you switch between these 2 rows, we believe that this would be switching because of introgression. Okay? And again, in the more general case where we have more reticulations, then you can have multiple rows and more switching. And then when we have this formulation, then the problem of interest is solved really by looking at the posterior decoding, which we can be solved with the forward and backward algorithms. So now the probability that a site J is coming on in local genealogy Tx and from parental species tree psi Y is basically obtained using the posterior decoding. So we have implemented all of this, and we have analyzed a set of mouse genomes. And the genomes that we look at here are two mouse species. One is called Mus spretis, and the other one is Mus musculus domesticus. And the reason we focus on this is because, as I showed in the beginning, the study by Michael Cohen and his colleagues, where they showed this introgression, adaptive introgression involving VCOR-C1 gene, was happening actually between Mus spretis and Mus domesticus. But there, they just focused on one region and they investigated it experimentally. These results here show our scan of introgression in chromosome 7 in mouse. And this is the region that's highlighted here is a region where VCOR-C1 is. So our method detected this region, and it's actually the largest region that it detected to be introgressed, and VCOR-C1 is here. And there are actually a few more that we detect. And the results can be summarized. We found 19 regions in the genomes that are introgressed. 5 of them are longer than 1 megabase pairs, and 14 of them are shorter than that. The length matters. It's very important. It's a very powerful signal. Actually, you can talk about the age of introgression and so on. One of the interesting things is actually we found that all the introgression segments to be concentrated in 5 chromosomes. I think the mouse has about 19 chromosomes. We found absolutely no signal of introgression in the other ones. Okay. So to summarize, if we view phylogenetic network as a collection of these MUL-TREE and allele mapping pairs, this provides us with a natural way to extend the multi-species coalescent for computing gene probabilities and ILS and hybridization. Also, this allows us to extend the hidden Markov models to annotate genomes in the presence of introgression as well. A major challenge now for us is definitely computational requirements. So I cannot do this hidden Markov model approach on 10 genomes, for example, or even the inference of networks, we cannot do it on 50 taxa at this point. All these methods, everything that's described is implemented. It's open source in Java, and it's publicly available. And just one last summarizing concept that I want to emphasize is that there is this I would say tension between the tree camp and the network camp, and there are more papers appearing recently talking about network thinking and so on. I believe that the truth lies somewhere in between them. The thing is that if you stick with methods that construct trees, you are going to get a tree, whether you like it or not, even if there's tons of hybridization. But the problem with networks also, and I think this has been stopping the community from adopting them, the problem with networks is that you can keep going to the right as much as you want. And this is actually actual analysis of the YEAST data set with 106 loci. If you invoke every incongruence there is because of reticulation, you will get a very complex picture that's not easy for biologists to accept. Right? But if you go somewhere in between and you develop a model that will incorporate both, this is what we obtain, actually. There's still a lot of the incongruence is due to incomplete lineage sorting, but we infer, actually, that many of the branches are too long for incomplete lineage sorting to be the issue. Instead, it is introgression. Okay. And this has been work with James Dignan from University of Canterbury in New Zealand, and with my group members at Rice, and I acknowledge the funding sources, and I thank you for your attention.
Can you clarify what you're meaning by a hybridization? Are you really thinking of two cells of different species hybridizing and starting with equal numbers of both?
Yeah.
And is that the same as, and so what's the difference between that and introgression?
Oh, so for me, I'm a computer scientist, so if there are biologists here, they can correct me. So hybridization is the event that happens between the species. For example, again, we have sexual reproduction between 2 members from 2 species, 2 populations, and so on. Then you have, again, that individual with, 50% material from the parents. Introgression, to me, the way I look at introgression, or I understand introgression, it is the integration of genetic material in the genome, right? So we are looking at extant genomes today. You can have hybridization, and because of no non-adaptive, no adaptation, because of drift and so on, you might after some generations, you might not see anything in the genome left.
but it doesn't necessarily generate a new species. So
No. Here we are not talking.
New species are generated because they're hybrids of the.
No. That's generating a new species is hybrid speciation, which is a special case of hybridization. Here I'm not talking about hybridization giving rise to a new species, and this is why I tried even to draw it. I mean, the drawing is not going to make any difference inside in terms of the data structure or the representation. But usually, we try to differentiate between hybrid speciation in the drawing. So this will be hybrid speciation in the sense that both are equal contributors, and this is the hybrid species. And usually, you'll see something like this, some sort of gene flow happening from this population to this. And again, the genetic material is going to be part of it is coming from here, part of it coming from here, but this is not a new species. But hybrid speciation does happen, but not every hybridization event is going to give rise to hybrid speciation.
So your model assumes these hybridization events as really as discrete events in time, right? So, but if in reality you have a very low rate of G flow between two species, what kind of pattern would that result in your model?
So, very good question, because this is sometimes I get this question about how does this differ, for example, from isolation with migration and all the work that models gene flow explicitly. So this is a very abstract model. We are not going after gene flow the rates or anything like that. But when we do the inference, when we are searching the network space, nothing stops our procedure. And in fact, our implementation allows for this kind of scenario. Right? It's going to allow him because these are two at different times and so on. So you can imagine that different hybridization events happening here, back crossing and so on. These are allowed by the search. And this is why I said that the space of phylogenetic network is actually infinite, because I can keep even partitioning this more and more. So we do allow this. It is still discretized in some sense. We are not saying, I'm not imagining it here like there's a tube here and again gene flow happening continuously within that.
But you would somehow discount for this, right? So that if you have very many hybridization events, and then there should be some statistical way to weigh this down in your framework?
Like going in favor of this instead of this?
Yes.
No, you don't have to. But this is where I put one slide in passing and did not talk about it in detail. So there is, if you start thinking about the more refined model where you look at the gene trees with their branch lengths and so on, I do believe that gene trees with branch lengths are going to give you more power, and you're going to be able to detect more of these scenarios than gene tree topologies alone. Right? And the only reason that currently we don't focus on that in the implementation is because I believe it's very hard to estimate these branch lengths. I mean, we are already dealing with hybridization and incomplete lineage sorting and error in the gene tree estimates. And if we put branch lengths into the picture, that's a fourth source of error as well. So I don't see our model taking this in favor of this. It all depends on the likelihood, and if we use cross-validation, which one gives us a better predictive power in some sense. But in our implementation, no, we do not have to favor this in place of this.
I feel like I understand your cross-validation application, but can you say a little bit about how you parameterize for AIC and BIC?
I mean, for AIC, for example, AIC and BIC, we look at what are the parameters in the phylogenetic network. For us, it is the number of branches, because they're going to give us branch lengths, each one of them. And for these nodes in degree 2, we add one parameter for each because that's the gamma. So this will be the number of parameters. For BIC, what we are using also for the number of samples is the number of gene trees in this case.
I mean, does that not penalize the one on the left?
This one?
Yeah.
Because of the likelihood is in the formula. I mean, you will have the penalty coming from the number of parameters, but you add one more parameter, and then your likelihood might drop significantly. In fact, actually, I am not coming here to advocate AIC or BIC.
Right. I think what Joachim was saying is that, like, you could have like a low rate of gene flow in. It's still just one parameter. It's just a different parameter.
Yeah.
And so if you want to model that, you have to do that with many parameters, and so it looks Like
Yeah, but we don't. Again, this is a much more abstract than all these approaches that take very population genetic approach and go after these rates and the different rates as well.
Okay, so if there's no more questions,
Thank you.
we have